All right, let's jump right in. Today, we're on a mission to crack one of the most fundamental codes in all of biology, the protein code. We're going to see how scientists go from this chaotic soup of molecules, to a single pure protein, and then how they read its secret message, letter by letter. It all starts with a problem that looks something like this. What you're looking at is basically the chaos inside a single cell. Every single one of those little spots, that's a different protein. And there are thousands of them, all jumbled up together doing their thing. They're the workers, the messengers, the very building blocks of life. So that leads to the million-dollar question for scientists, right? In that massive molecular crowd, how on earth do you find and pull out just one specific protein you want to study? It's like trying to find one specific person in a stadium packed with thousands of people. Where do you even begin? Well, you treat it like a detective story. Seriously, it's a mission. It requires a stakeout to isolate our target, then a lineup to confirm its identity, and finally a full-on interrogation to get it to spill its secrets. And that's the exact journey we're going on right now. Okay, so first things first. Let's really get a handle on the sheer scale of this protein puzzle. Why is this mission so critical? And just how tough is it really? To put a number on it, get this. Even a quote-unquote simple organism like an E. coli bacterium, the workhorse of biology labs, has over 3,000 different types of proteins inside it. A human cell, that number explodes into the tens of thousands. The haystack is absolutely enormous, and our needle is microscopic. And here's why it matters. This isn't just some abstract scientific puzzle. As this quote makes crystal clear, thousands of genetic diseases, things that affect real people, are caused by a single protein going haywire. So if we ever want to understand and fight these diseases, we have to find that one faulty protein first. The stakes just couldn't be higher. So we've got our mission. It's time for the stakeout. The first step is all about purification. We need a way to keep an eye on our target protein as we start filtering out all the molecular junk we don't care about. And to do that, scientists use two key metrics. First, there's total activity. That one's pretty straightforward. It's just how much of our target protein we have in total. But the real clever one, the one that tells us if we're getting warmer, is specific activity. This measures the purity, the proportion of our target protein compared to everything else in the mix. Let me break that down with an analogy. Imagine you have two big buckets of marbles. Both buckets have exactly 10 red marbles. That's our total activity, and it's the same in both. But look, the second bucket has way fewer other colored marbles, right? That means the proportion of red marbles, the specific activity, is much, much higher. Every step of purification is like reaching in and pulling out a handful of non-red marbles. The total number of red ones might go down a tiny bit, but their purity, their specific activity, just skyrockets. And that's how we know we're closing in on our target. Okay, so we've run our purification steps. Our specific activity is through the roof. We think we've got our guy, but how do we know for sure? How do we confirm the identity? Well, now it's time for the molecular lineup. And this is the brilliant technique they use to do it. It's this super clever two-step process. First, they take the whole protein mix and separate it horizontally based on each protein's electrical charge. Then they take that line of proteins, turn it 90 degrees, and separate it again, but this time vertically based on size. It's kind of like sorting a deck of cards, first by suit and then sorting each suit by number. And the result is this. It's absolutely stunning. Every single one of those dots is a different type of protein, perfectly separated from all its neighbors. Now, if our purification worked, we should see one spot that's way bigger and darker than all the others. That's our target. This is the moment of truth in our lineup. So we've found our protein, we've confirmed it's pure. Now comes the most critical part of the entire mission, the interrogation. It's time to find out its primary structure, its unique sequence of amino acids, the very letters that spell out what that protein is and what it does. And to really appreciate how amazing our modern tools are, just look at this. Back in 1953, the legendary Frederick Sanger was the first person to ever do this with the hormone insulin. It was a monumental, Nobel Prize-winning achievement that took him 10 years. 10! Today, that same job can be done in a day or two. That's just mind-blowing progress. So how does it actually work? Well, the modern method is called the Edman degradation, and it's this beautifully elegant, repetitive loop.
A machine grabs the long protein chain, slaps a special chemical label onto the very first amino acid at the end, and then just snips it off. Then it identifies that one amino acid, records what it is, and does the whole thing over again on the new end of the chain. Label, cleave, identify. Over and over and over.
But here's the catch, and it's a huge one. For this to work for dozens of cycles in a row, each step has to be practically perfect, more than 99% efficient. It's an incredible display of precision chemistry. If it were any less, the signal would get messy and scrambled after just a few rounds. But at this level of accuracy, you can read a sequence of 50 or more amino acids. Easy. So, mission accomplished. We've cracked the code. We have our long string of letters. But let's bring it all home. So what? Why does this simple sequence matter so much?
Because, and this is the absolute key to everything, it all flows from that sequence. That sequence of amino acids dictates exactly how the protein folds up into a complex 3D shape. That unique 3D shape is what dictates the protein's specific job in the body. So a single error, one wrong letter in that sequence, can change the shape. That altered shape can break the protein's function. And that broken function, that can lead directly to disease. It's a straight line from the code to our health.
So you see, every protein sequence that scientists uncover isn't just data. It's a story. It's a biological story of evolution, of function, and sometimes a story of disease. And with the incredible tools we have today, the biggest question isn't, can we read the story? It's, which of the thousands of unread stories out there will we decide to crack open next?